This video is presented by Subi, lecturer from Emmanuel Arashar College of Pharmacy. Today I am going to teaching about dissolution. Dissolution is the process by which a solid substance is dissolved in a liquid to form a solution. What is meant by dissolution? Dissolution is the process by which a solid substance is dissolved in a liquid to form a solution. Next, dissolution test. It is an important quality control test used to study the rate and extent of drug release from solid dosage form like tablet and capsules. It is an important quality control test used to study the rate and extent of drug release from solid dosage forms like tablets and capsules. This is one of the official test or pharmacopoeial test. Next, move on. Types of dissolution apparatus. There are mainly two commonly used types of dissolution apparatus are basket type and paddle type. Basket type uses a rotating basket. Tablets or capsule is placed inside the basket. In basket type apparatus, using rotating basket and tablet or capsule is placed inside the basket. Next, paddle type apparatus using a rotating paddle. Tablet is placed at the bottom of the vessel. Paddle type apparatus using a rotating paddle and tablet is placed at the bottom of the vessel. First of all, see about dissolution apparatus, that is basket type dissolution apparatus. Definition of basket type dissolution apparatus. Basket type dissolution apparatus is an instrument used to determine the rate of drug dissolution from solid dosage forms like tablets and capsule using a rotating basket. Basket type dissolution apparatus is an instrument used to determine the rate of drug dissolution from solid dosage forms like tablets and capsules using rotating basket. Principle of basket type dissolution apparatus. The tablet is placed inside a Wehrmacht's basket which rotates in a dissolution medium like water or buffer. As the basket rotates, the drug dissolves into the medium. The samples are taken at intervals to measure the amount can be dissolved. The tablet is placed inside a Wehrmacht's basket which rotates in a dissolution medium like water or buffer. As the basket rotates, the drugs dissolve into the medium and samples are taken at intervals to measure the amount dissolved. This is the picture of basket type dissolution apparatus. Parts of basket type dissolution apparatus are vessels, dissolution medium, shaft, motor, water bath, thermometer and sampling port. What is the use of dissolution vessel? Dissolution vessel, it is one of the glass container made up of glass, usually having 1000 ml capacity. Dissolution vessel made of glass, it is having 1000 ml capacity. Then basket It is stainless steel mesh. This basket is made up of stainless steel mesh. It is used to hold the tablet. It is used to hold the tablet. Then rotating shaft connects motor to basket. Connects motor to basket. Motor provide constant rotation. It is provide constant rotation, usually 50 to 100 rpm. Water bath or heater maintain temperature at 37 degree. Its temperature maintained at 37 degree. Thermometer is to monitor the temperature. Thermometer is to monitor the temperature. Sampling fourth. For withdrawal the samples. It is used to withdrawing the samples at a particular interval time. Next move on. Procedure for dissolution basket type dissolution apparatus. First of all, fill the vessel with the dissolution medium, usually 900 ml. Maintain temperature at 37 degree. Place tablet inside the basket. Rotate basket at a specified speed. That means rotate the basket at a specific speed. Withdraw samples at regular intervals. Analyze drug contents using UV or other methods. This basket type apparatus consists of cylindrical stainless steel basket made up of wire with aperture size of 425 micrometer attached to the disc on the riving shaft. The basket consists of two components. The top part with a vent is attached to the shaft. The lower part is detachable, which is made up of stainless steel. For placing the tablet, it is immersed in 9000 ml of dissolution medium. Uses of basket type dissolution apparatus. Test and drug release rate from tablets and capsule. Quality control in pharmaceutical industries. Comparing different formulations. Next move on. Advantages and disadvantages of basket type dissolution apparatus. Advantages: suitable for floating dosage forms, prevent tablet from sticking to vessel bottom, uniform exposure to dissolution medium. Disadvantages: not suitable for disintegrating tablets. Air bubbles can affect result. Air bubbles can affect results. Next move on. Paddle type dissolution apparatus. Paddle type dissolution apparatus. Definition: paddle type dissolution apparatus is used to determine the rate of drug dissolution from tablets and capsules using a rotating paddle immersed in dissolution medium. Paddle type dissolution apparatus is used to determine the rate of drug dissolution from tablets and capsules using a rotating paddle immersed in dissolution medium. Principle of paddle type dissolution apparatus. The tablet is placed at the bottom of the vessels and a paddle rotates above it. The rotation creates uniform mixing, allowing the drug to dissolve into the medium. Samples are taken at intervals to measure dissolution. The tablet is placed at the bottom of the vessels and a paddle rotates above it. The rotation creates uniform mixing, allowing the drug to dissolve into the medium. Samples are taken at intervals to measure dissolution. Parts of paddle type dissolution apparatus. Dissolution vessel. is a cylindrical vessel. It is made up of glass or other transparent material having 1000 ml capacity. It is fitted with a lid having 4 holes. It is fitted with a lids having 4 holes, one for soft top stirrer, second for placing the thermometer, and remaining 2 for sample removal. Paddle. Paddle, flat blade attached to shaft. It is attached to shaft. Rotating shaft connects paddle to motor. Rotating shaft. It connects paddle to motor. Then motor. Control speed. An electrical motor with speed regulator capable of maintaining the speed, rotating a paddle or basket. An electric motor with the speed regulator capable of maintaining the speed of rotation of paddle or basket. Water bath. To maintain 37 degree. Why 37 degree maintains? It means body temperature. Yes. Thermometer used to check the temperature. Sampling port used to withdraw sample at regular interval. Suitable device for withdrawal of sample and water bath with the temperature control inside the vessel, which will maintain at 37 degree. Next, procedure for paddle type dissolution apparatus. Place 900 ml of dissolution medium into the vessel. Warm the dissolution medium to 37 degree. Place one tablet in the each basket or paddle of the apparatus. Place one tablet in the each basket or paddle of the apparatus. Operate the apparatus at the speed of rotation specified in the monograph. The samples are withdrawn at a frequent interval as mentioned in the monograph, and the volume taken is replaced with fresh dissolution medium to maintain the sink condition. The sample is filtered and analyzed as per monograph to determine the amount of APA dissolved in solution. Analyze recommended using UV or spectrophotometer. Place 900 ml of dissolution medium into the vessel, maintaining temperature at 37 degree. Place tablet at the bottom of the vessel. Rotate paddle at a specified speed. Withdraw a sample at a fixed time intervals. Analyze drug content using UV or spectrophotometer. Uses of paddle type dissolution apparatus. It is most widely used for tablet dissolution testing, suitable for immediate release dosage form. It is suitable for immediate release dosage form. It is used to quality control in pharmaceutical industry. Advantages of paddle type dissolution apparatus. It is easy and simple to operate, suitable for most tablet, no mess clogging, unlike basket type. It is easy to simple to operate, suitable for most tablets, no mess clogging, unlike basket type. It's having some disadvantages. They are: tablet may float or stick to vessel bottom. Tablets may be float or stick to vessel bottom. Not suitable for floating dosage form. It is not suitable for floating dosage form. Next, move on non-official test or non-pharmacopoeial test. Harness test. Appearance, thickness and friability. Appearance, harness, thickness and friability. These are all non-pharmacopoeial or non-official test. Let's see one by one. First, general appearance. Pharmaceutical elegance is important for consumer acceptance of tablet. Pharmaceutical elegance is important for consumer acceptance of tablet. The control of general appearance involves evaluating the following parameters. They are size, shape, color, odor, taste. They are size, shape, color, odor, taste. Color. The color of tablets should be uniform within a single tablet, between tablets, from batch to batch, that means lot to lot. The color of tablets should be uniform within a single tablet, between tablets, or from batch to batch, or lot to lot. Odor. The presence of an unusual odor may indicate instability or degradation of the drug. Odor. The presence of an unusual odor may indicate instability or degradation of the drug. Taste. Tablets should have an acceptable taste, especially for chewable or disposable tablets. Tablets should have an acceptable taste, especially for chewable or disposable tablets. Then, shape and size should be consistent for easy identification and patient compliance. It is easy to identify and patient compliance. Next, move on tablet corners, that means crushing strength. Harness is the force required to break a tablet. Harness is the force required to break a tablet. It is also known as tablet crushing strength. Some testers are used to identify tablet harness. They are Monsanto harness tester, Pfizer harness tester, Strongbow testers, Ervica tester. Monsanto harness tester, Pfizer harness tester, Strongbow tester, and Ervica tester. Monsanto harness tester. Place the tablet between the spindle and anvil, that means across its diameter. Adjust the knob to hold the tablet firmly. To adjust the knob to hold the tablet firmly. Set the pointer to 0 using the adjusting screw. Apply force by rotating the screw slowly. Continue till the tablet breaks. Note the reading on the scale. Place the tablet between the spindle and anvil. Adjust the knob to hold the tablet firmly. Set the pointer to 0 using the adjusting screw. Apply force for rotating the screw slowly. Continue until the tablet breaks. Note the reading on the scale. It is expressed in kilogram per centimeter square. Kilogram per centimeter square. Acceptable range is 4 to 8 kg per centimeter square is considered suitable for most tablet. Ensure Monsanto harness tester used to ensure mechanical strength of tablets, prevents packaging during handling and transport, affects disintegration and dissolution. Ensure mechanical strength of tablets, prevents breakage during handling and transport, affects disintegration and dissolution. Next, move on tablet thickness. Thickness of a tablet is measured using a micrometer or vernier caliper and is expressed in millimeters. Importance. Ensure uniformity of tablet size. Important for packaging. Helps maintain dose consistency and appearance. Importance of tablet thickness and ensure uniformity of tablet size. Important for packaging. Helps maintaining dose consistency and appearance. Next we move on friability of tablets. Friability is the friability is the test used to evaluate the ability of tablets to withstand wear and tear during packaging, handling and transportation. Friability is the test used to evaluate the ability of tablets to withstand wear and tear during packaging, handling and transportation. Apparatus used to friability test are Roach friability. It is consist of a plastic rotating drum. It's rotated at 25 rpm. It is consist of plastic rotating drum. Rotate at 25 rpm. Procedure. It is performed to evaluate the ability of the tablet to withstand wear and tearing in packaging, handling and transporting. Roach friability apparatus is used to measure the friability. It is consist of circular plastic chamber with rotates at a speed of 25 rpm. 20 tablets are weighed and placed in a plastic chamber. First 20 tablets are weighed and placed in a plastic chamber. The chamber is rotated for 4 minutes or 100 revolutions. Rotating speed 25 rpm. The chamber is rotated for 4 minutes or 100 revolutions. Rotating speed 25 rpm. During each revolution, the tablet falls from a distance of 6 inches. During each revolution, the tablets falls from a distance of 6 inches. The tablets are removed from the chamber after 100 revolution. The tablets are removed from the chamber after 100 revolutions and weighed. Loss in weight indicates the friability. Loss in weight indicates the friability. Percentage friability can be calculated from the difference between these two weights. Percentage friability can be calculated from the difference between these two weights. Percentage friability should not be more than 1%. Percentage friability. W1 minus W2 divided by W1 into 100. This is the formula of calculation of percentage friability. Acceptable limit. Friability should not more than 1%. It is should not more than 1%. Importance of ensure mechanical strength of tablet, prevents breakage and powdering, maintains product quality. Ensure mechanical strength of tablet, prevents breakage and powdering, maintains product quality. Okay, today lesson is over. See you next class. Thank you.